living matter of cell, that is protoplasm, consists of both organic and inorganic compounds. Various types of organic and inorganic materials are found in a cell. The molecules present in the living organisms are called biomolecules. These molecules combine to form inorganic material and organic compound. Of all the inorganic materials, water is the most important. Other inorganic substances are salt and minerals. You know, organic compounds include carbohydrates, lipids, amino acids, proteins, nucleotides, nucleic acids, vitamins, etc. These biomolecules play an important role in the structure and functions of living organisms. Out of these, proteins are very important. You must be knowing that different kinds of reactions taking place in a body are catalyzed by different kinds of enzymes, which are basically proteins.
Students, ਅੱਜ ਅਸੀਂ proteins ਦੇ structure, types ਅਤੇ functions ਬਾਰੇ ਪੜਾਂਗੇ. Students, protein word ਗ੍ਰੀਕ ਭਾਸ਼ਾ ਦੇ ਸ਼ਬਦ proteos ਤੋਂ ਲਿਆ ਗਿਆ ਹੈ, ਜਿਸ ਦਾ ਅਰਥ ਹੈ first order. Proteins ਸਾਡੇ ਸਰੀਰ ਦੇ basic structure ਅਤੇ functions ਵਿੱਚ ਮਹੱਤਵਪੂਰਨ ਰੋਲ ਅਦਾ ਕਰਦੇ ਹਨ. Proteins are nitrogenous compounds containing carbon, hydrogen, oxygen and nitrogen. Basically, proteins are made up of amino acids. They are the structural framework of all cell components. They also play an important role in cell division and heredity. As you know, proteins are essential components of protoplasm. In today's lesson, we will discuss about the structure of proteins, their types and functions. But before going into details, first let me share the learning objectives of today's topic.
On completion of this topic, learners will be able to list the components of proteins, discuss chemical structure of proteins, describe different structural levels of proteins, classify the various types of proteins, discuss the functions of proteins.
ਬੱਚਿਓ, proteins ਉਹ ਮੈਕਰੋਮੋਲੀਕਿਊਲਸ ਹਨ ਜਿਹੜੇ ਕਿ ਅਮੀਨੋ ਐਸਿਡਸ ਤੋਂ ਮਿਲ ਕੇ ਬਣਦੇ ਹਨ. ਇਹ ਪ੍ਰੋਟੀਨ ਕਾਰਬਨ, ਹਾਈਡਰੋਜਨ, ਆਕਸੀਜਨ, ਨਾਈਟ੍ਰੋਜਨ ਅਤੇ ਸਲਫਰ ਨਾਲ ਮਿਲ ਕੇ ਬਣੇ ਹੁੰਦੇ ਹਨ. ਸਾਰੇ ਪ੍ਰੋਟੀਨ ਅਮੀਨੋ ਐਸਿਡ ਦੇ ਪੋਲੀਮਰ ਹਨ. ਪ੍ਰੋਟੀਨ ਸ਼ਬਦ ਬ੍ਰਾਜ਼ੀਲੀਅਸ ਅਤੇ ਮੋਲਡਰ ਨੇ 1838 ਵਿੱਚ ਗ੍ਰੀਕ ਵਰਡ ਪ੍ਰੋਟੀਓਸ ਤੋਂ ਡਿਰਾਈਵ ਕੀਤਾ. ਬੱਚਿਓ, ਸਾਰੇ ਲਿਵਿੰਗ ਆਰਗੇਨਿਜ਼ਮਸ ਦੇ ਸਟਰਕਚਰ, ਬਿਹੇਵੀਅਰ ਅਤੇ ਕੁਆਲਿਟੀਜ਼ ਉਨ੍ਹਾਂ ਵਿੱਚ ਮੌਜੂਦ ਪ੍ਰੋਟੀਨਸ ਤੇ ਨਿਰਭਰ ਕਰਦੇ ਹਨ. ਪ੍ਰੋਟੀਨਸ ਸੈਲ ਕੰਟੈਂਟ ਦਾ 12% ਹਿੱਸਾ ਫਾਰਮ ਕਰਦੇ ਹਨ.
Proteins are an important class of biological macromolecules present in organisms. All proteins are polymers of amino acid. Polymers are also known as polypeptides. They consist of sequence of 20 different alpha amino acids. As proteins have to perform several biological functions, they take different shapes by folding themselves. These folding are possible by a number of non-covalent interactions such as hydrogen bonding, ionic interactions, Vanderwaal's forces and hydrophobic packing.
Students, proteins amino acids ਦੀ ਕੰਡੈਂਸੇਸ਼ਨ ਤੋਂ ਬਣਦੇ ਹਨ. ਪ੍ਰੋਟੀਨਸ ਦੇ ਸਟਰਕਚਰ ਬਾਰੇ ਪੜ੍ਹਨ ਤੋਂ ਪਹਿਲਾਂ ਆਓ ਅਸੀਂ ਅਮੀਨੋ ਐਸਿਡ ਦੇ ਬੇਸਿਕ ਸਟਰਕਚਰ ਬਾਰੇ ਜਾਣੀਏ. ਅਮੀਨੋ ਐਸਿਡ ਦੇ ਇੱਕ ਸਮਾਲ ਮੋਲੀਕਿਊਲ ਨੂੰ ਮੋਨੋਮਰ ਕਿਹਾ ਜਾਂਦਾ ਹੈ। ਅਮੀਨੋ ਐਸਿਡ ਦੇ ਇੱਕ ਮੋਲੀਕਿਊਲ ਵਿੱਚ ਸੈਂਟਰਲ ਐਲਫਾ ਕਾਰਬਨ ਐਟਮ ਹੁੰਦਾ ਹੈ। ਇਸ ਸੈਂਟਰਲ ਕਾਰਬਨ ਐਟਮ ਦੇ ਇੱਕ ਪਾਸੇ ਅਮੀਨੋ ਗਰੁੱਪ ਅਤੇ ਦੂਜੇ ਪਾਸੇ ਕਾਰਬੋਕਸਿਲ ਗਰੁੱਪ, ਤੀਜੇ ਪਾਸੇ ਹਾਈਡਰੋਜਨ ਐਟਮਸ ਅਤੇ ਚੌਥੇ ਪਾਸੇ ਇੱਕ ਸਾਈਡ ਚੇਨ ਹੁੰਦੀ ਹੈ। ਇਹ ਸਾਈਡ ਚੇਨ ਪੋਲਰ ਵੀ ਹੋ ਸਕਦੀ ਹੈ ਅਤੇ ਨਾਨ ਪੋਲਰ ਵੀ।
Structure of proteins. The monomers of amino acid join with each other to form chain-like macromolecules called polymers. These macromolecules, having high molecular weight, are called protein. In a molecule of protein, amino group of one amino acid combines with carboxyl group of its neighboring amino acid. In this whole process, a molecule of water is eliminated and a peptide NHCO bond is formed. I think it is now clear that proteins are polymers of amino acid. The structure of proteins was first described by German chemist Emil Fischer in 1900.
Students, ਜਦੋਂ ਦੋ ਅਮੀਨੋ ਐਸਿਡਸ ਇੱਕ ਪੈਪਟਾਈਡ ਬੌਂਡ ਨਾਲ ਜੁੜਦੇ ਹਨ ਤਾਂ ਉਸ ਨੂੰ ਡਾਈਪੈਪਟਾਈਡ ਕਹਿੰਦੇ ਹਨ। ਇਸ ਤਰ੍ਹਾਂ ਤਿੰਨ ਅਮੀਨੋ ਐਸਿਡਸ ਦੇ ਸੀਕੁਐਂਸ ਨੂੰ ਟ੍ਰਾਈਪੈਪਟਾਈਡ ਕਿਹਾ ਜਾਂਦਾ ਹੈ। ਇੱਕ ਪੋਲੀਪੈਪਟਾਈਡ ਵਿੱਚ ਅਨਸਪੈਸੀਫਾਈਡ ਨੰਬਰ ਆਫ ਅਮੀਨੋ ਐਸਿਡਸ ਹੁੰਦੇ ਹਨ, ਪਰ ਜਨਰਲੀ 20 ਤੋਂ ਜ਼ਿਆਦਾ ਅਮੀਨੋ ਐਸਿਡਸ ਹੁੰਦੇ ਹਨ।
Organizational or structural levels of proteins. Well, you all know that proteins are among the most complex of all organic compounds. 4 basic structural levels of protein have been proposed, considering the relative dimensions of the protein molecules. These structural levels are primary structure, secondary structure, tertiary structure, quaternary structure. Primary structure. The linear sequence of different amino acids is known as primary structure of the protein molecule. The sequence of amino acid is determined by the sequence of nucleotides of DNA of cell's nucleus. The work of finding out amino acid sequence of protein was first pioneered by the double Nobel Prize winner Frederick Sanger and his colleagues. They were successful in determining the primary structure of insulin, the first protein of which primary structure was explained. Insulin is a pancreatic hormone consisting of 51 amino acid units. You know, sequence of amino acids in a protein chain would determine its physical, biological, and chemical properties. Secondary structure. Pauling and Corey, by X-ray diffraction studies, found that the polypeptide chains are not straight but are folded and coiled to become orderly and stable elements of protein. This is called the secondary level of organization. The helical structure is maintained by a series of regularly spaced hydrogen bond. This causes the peptide to twist or fold into 2 basic types: alpha helix, B-Pleated sheets.
Students, ਜਦੋਂ ਪੋਲੀਪੈਪਟਾਈਡ ਚੇਨ ਇੱਕ ਕੋਇਲ ਦੀ ਤਰ੍ਹਾਂ ਅਰੇਂਜਡ ਹੁੰਦੀ ਹੈ ਤਾਂ ਉਸ ਨੂੰ ਐਲਫਾ ਹੈਲਿਕਸ ਸਟਰਕਚਰ ਕਿਹਾ ਜਾਂਦਾ ਹੈ। ਇਸ ਹੈਲਿਕਸ ਵਿੱਚ ਅਮੀਨੋ ਐਸਿਡਸ ਇਸ ਤਰ੍ਹਾਂ ਪਏ ਹੁੰਦੇ ਹਨ ਕਿ ਉਹਨਾਂ ਦੀਆਂ ਸਾਈਡ ਚੇਨਸ ਸਪਾਇਰਲ ਦੇ ਬਾਹਰ ਵੱਲ ਹੁੰਦੀਆਂ ਹਨ। ਐਲਫਾ ਹੈਲਿਕਸ ਇੱਕ ਟਾਈਟਲੀ ਕੋਇਲਡ ਸਟਰਕਚਰ ਹੈ ਜਿਸ ਦੀ ਬਣਤਰ ਇੱਕ ਸਰਕੂਲਰ ਪੌੜੀ ਨਾਲ ਮਿਲਦੀ ਜੁਲਦੀ ਹੈ। ਐਲਫਾ ਹੈਲਿਕਸ ਸੈਕੰਡਰੀ ਸਟਰਕਚਰ ਕੈਰੇਟਿਨ ਨਾਂ ਦੇ ਪ੍ਰੋਟੀਨ ਵਿੱਚ ਪਾਈ ਜਾਂਦੀ ਹੈ। ਇਹ ਉਹ ਪ੍ਰੋਟੀਨ ਹੈ ਜੋ ਕਿ ਹੇਅਰ, ਫਰ, ਵੂਲ, ਕਲੌਜ਼, ਹੌਰਨਸ, ਹੂਵਸ, ਫੈਦਰਸ etc. ਨੂੰ ਬਣਾਉਂਦੀ ਹੈ।
Beta-pleated sheet structure. When two or more chains are attached together by intermolecular hydrogen bonds, it is called beta-pleated sheet structure. In this, peptide chains can be parallel or antiparallel. In a parallel chain, beta-pleated sheet, nitrogen atoms of adjacent strands of polypeptides point in the same direction. Example is beta-carotene. In an anti-parallel chain, beta-pleated sheet, nitrogen atoms of adjacent strands of polypeptides point in opposite direction. The example is silk fiber. Students, you know antiparallel beta-pleated sheets are more stable. Tertiary structure. It involves bending and folding of alpha helix or beta-pleated peptide chains upon themselves to assume a complex but specific forms like spherical, rod, etc. These geometrical or three-dimensional structures are known as tertiary structure of proteins. The tertiary structure of a protein brings distant amino acid side chains nearer to the active sites of enzyme proteins. The tertiary structure is maintained and stabilized by three types of bond. Ionic bond. Such bonds are formed due to attraction of forces between oppositely charged ionized groups. Examples are NH3 and COO. Hydrogen bond. These bonds are formed due to sharing of hydrogen proton by two electronegative atoms. Disulfide bond. It is formed between two amino acids containing sulfur as cysteine.
Students, out of these bonds, disulfide bonds are the strongest of all the bonds. The tertiary structure can be broken by high energy radiations, high temperature, fluctuations in pH, which leads to denaturation of proteins. Quaternary structure. The structure formed by several protein molecules, that is two or more polypeptide chains, is the quaternary structure. Hemoglobin, insulin and liver protein have quaternary structure. Hemoglobin has 4 helical polypeptide chains, 2 alpha and 2 beta chains. Insulin is composed of 2 polypeptide chains linked by sulfur bridges. Students, liver protein are formed of 20 identical polypeptides. ਬੱਚਿਓ, ਹੁਣ ਤੁਹਾਨੂੰ ਪਤਾ ਲੱਗ ਹੀ ਗਿਆ ਹੈ ਕਿ ਪ੍ਰੋਟੀਨਸ ਬਹੁਤ ਵੱਡੇ ਮੋਲੀਕਿਊਲਸ ਹਨ। ਬੱਚਿਓ, ਤੁਹਾਨੂੰ ਇਹ ਵੀ ਪਤਾ ਹੋਣਾ ਚਾਹੀਦਾ ਹੈ ਕਿ ਪ੍ਰੋਟੀਨਸ ਦੀ ਨੇਚਰ ਐਮਫੋਟੇਰਿਕ ਹੁੰਦੀ ਹੈ ਕਿਉਂਕਿ ਇਹਨਾਂ ਦਾ ਅਮੀਨੋ ਗਰੁੱਪ ਬੇਸਿਕ ਹੁੰਦਾ ਹੈ ਅਤੇ ਇਹਨਾਂ ਦਾ ਕਾਰਬੋਕਸਿਲ ਗਰੁੱਪ ਐਸਿਡਿਕ ਹੁੰਦਾ ਹੈ ਤੇ ਇਹ ਬੇਸਿਸ ਨਾਲ ਕੰਬਾਈਨ ਹੋ ਸਕਦਾ ਹੈ। ਇਸ ਐਮਫੋਟੇਰਿਕ ਨੇਚਰ ਦੇ ਕਾਰਨ ਪ੍ਰੋਟੀਨਸ ਸੈਲਸ ਦੇ ਅੰਦਰ ਬਫਰ ਦੀ ਤਰ੍ਹਾਂ ਕੰਮ ਕਰਦੇ ਹਨ।
Classification of proteins. Proteins can be classified on the basis of components of molecules, functions, shape, and nature of molecules. On the basis of components of molecules, on the basis of composition, the protein can be simple proteins, conjugated proteins, derived proteins. Let us first discuss these types one by one. Simple proteins are made up of amino acids only. Additional non-amino groups are absent. The proteins which yield alpha amino acids only on hydrolysis are known as simple proteins. For example, histones and keratin. Conjugated proteins. These proteins yield alpha amino acids and a non-protein material on hydrolysis. The non-protein material is known as the prosthetic group. Depending upon the nature of prosthetic group, they can be glycoprotein, those having carbohydrate in association with protein molecule are glycoproteins. Examples are mucin of saliva, egg albumin, etc. Phosphoprotein. Prosthetic group attached is phosphoric acid. Example are casein of milk, vitelin of egg yolk. Lipoprotein. Prosthetic group is mainly phospholipids or cholesterol. Examples are proteins of blood serum, milk and egg yolk. Nucleoprotein. Prosthetic group is nucleic acid. Examples are deoxyribonucleoproteins of chromosomes and ribonucleoproteins of ribosomes. Metalloprotein. Prosthetic group is some metal ion. Examples are iron in hemoglobin and myoglobin, copper and magnesium in cytochromes. Third is derived proteins. Such proteins are the proteins produced due to partial hydrolysis of natural proteins. They can be of primary or secondary types. Primary derived proteins. They are derivatives of proteins where the size of protein molecule is not generally altered, such as proteosis. Secondary derived proteins. Derivations of natural protein formed due to hydrolysis. The molecules are smaller than the original protein molecules. They are peptones and polypeptides.
On basis of shape of their molecules. Composition ਤੇ based protein ਦੀ classification ਪੜਨ ਤੋਂ ਬਾਅਦ ਹੁਣ ਅਸੀਂ ਪੜਾਂਗੇ ਕਿ shape ਦੇ basis ਤੇ protein ਨੂੰ ਕਿੰਨੇ types ਵਿੱਚ divide ਕਰ ਸਕਦੇ ਹਾਂ। Shape ਦੇ basis ਤੇ proteins ਦੋ ਤਰ੍ਹਾਂ ਦੀਆਂ ਹੁੰਦੀਆਂ ਹਨ। Fibrous proteins। ਇਸ ਤਰ੍ਹਾਂ ਦੀਆਂ proteins ਵਿੱਚ polypeptides chains spirally twisted ਹੁੰਦੀਆਂ ਹਨ। ਇਹ ਪਾਣੀ ਵਿੱਚ insoluble ਹੁੰਦੀਆਂ ਹਨ। ਇਹ tough ਅਤੇ contractile ਵੀ ਹੁੰਦੀਆਂ ਹਨ। ਇਹਨਾਂ ਦੀਆਂ common examples ਹਨ collagen of connective tissue, actin and myosin of muscles, etc. Globular proteins। ਇਸ ਤਰ੍ਹਾਂ ਦੀਆਂ proteins ਵਿੱਚ polypeptide chains ਆਪਣੇ ਆਲੇ ਦੁਆਲੇ coil ਹੋ ਕੇ ਇੱਕ spherical molecule ਬਣਾ ਦਿੰਦੀਆਂ ਹਨ। ਇਹ ਆਮ ਤੌਰ ਤੇ non-contractile ਹੁੰਦੀਆਂ ਹਨ। ਇਹ enzymatic ਵੀ ਹੋ ਸਕਦੀਆਂ ਹਨ ਅਤੇ non-enzymatic ਵੀ। ਛੋਟੀਆਂ globular proteins ਪਾਣੀ ਵਿੱਚ soluble ਹੁੰਦੀਆਂ ਹਨ, ਜਦਕਿ ਵੱਡੀਆਂ globular proteins ਪਾਣੀ ਵਿੱਚ insoluble ਹੁੰਦੀਆਂ ਹਨ। ਇਹਨਾਂ ਦੀਆਂ examples ਹਨ egg albumin, hemoglobin of erythrocytes, glutens of wheat, etc.
On the basis of nature of molecules, proteins may be acidic or basic. They are acidic if they are rich in acidic amino acid, such as most blood proteins. They are basic if they are rich in basic amino acid, such as histone proteins associated with DNA. On the basis of their functions, on the basis of functions, proteins can be enzymatic proteins or non-enzymatic. Non-enzymatic proteins are of many types, like structural proteins. These form a part of cellular protein, like proteins of cartilage, skin, nails, horns, etc. Contractile proteins. These help in movement and locomotion, like proteins of skeletal muscles. Enzymatic proteins. These proteins function as enzymes and serve as biocatalysts in the chemical reactions, like enzymes secreted by different glands. Protective proteins protect the body against antigens like antibodies, which are responsible for immunity of the body. Storage proteins are found in the form of reserve food, mostly present in seeds, eggs, milk, etc., like albumin, globulins, gluten, etc. Hormones, like insulin, is also protein in nature.
ਬੱਚਿਓ, ਹੁਣ ਤੱਕ ਤੁਹਾਨੂੰ ਪਤਾ ਹੀ ਚੱਲ ਗਿਆ ਹੋਵੇਗਾ ਕਿ ਪ੍ਰੋਟੀਨਸ ਸਾਡੇ ਲਈ ਕਿੰਨੇ ਜ਼ਰੂਰੀ ਹਨ। ਪ੍ਰੋਟੀਨਸ ਦੀ ਸਟਰਕਚਰ ਐਂਡ ਟਾਈਪਸ ਡਿਟੇਲ ਵਿੱਚ ਪੜ੍ਹਨ ਤੋਂ ਬਾਅਦ ਹੁਣ ਅਸੀਂ ਪ੍ਰੋਟੀਨਸ ਦੇ ਫੰਕਸ਼ਨਸ ਬਾਰੇ ਪੜ੍ਹਾਂਗੇ। Functions of proteins. Let us discuss functions in detail. The proteins serve many functions, like proteins as structural components. Many proteins are the structural proteins for the formation of cell organelles like ribosomes, mitochondria, plastids, chromosomes, etc. Certain proteins also form horny layer of skin and exoskeleton structures like scales, feathers, hair, wool, nails, etc. Example is keratin. Proteins as fuel. Proteins are used as fuel to provide energy when carbohydrates and fats are in short supply. Role in defense. Antibodies or immunoglobulins form the immune system of the body to protect it from the foreign particles. Role in movement and locomotion. Certain proteins help in movement and locomotion of organisms. Example, actin and myosin present in muscles. Role in transport of materials. Many proteins serve as physical carriers of other functionally important smaller molecules like hemoglobin of red blood corpuscles, transports oxygen and carbon dioxide. Role in blood coagulation. Certain proteins cause clotting of blood at the time of injury and prevent blood loss, such as thrombin and fibrinogen. Monilin. It is a protein produced from an African berry. It is 200 times sweeter than sucrose. It is very useful for diabetic patients. I hope you all have enjoyed this topic on structure of proteins, types and functions. Let me summarize it for you. Proteins are polymers of amino acids. Proteins are made up of carbon, hydrogen, oxygen, nitrogen and sulfur. Protein word was derived from Greek word proteos by Berzelius and Mulder in 1838. Proteins consist of sequence of 20 different alpha amino acids. 4 basic structural levels of proteins have been suggested. This is primary structure, secondary structure, tertiary structure, and quaternary structure. Proteins can be classified on the basis of composition, functions, shape, and nature of molecules. On the basis of composition of molecules, proteins can be simple, conjugated, and derived proteins. On the basis of shape of their molecules, proteins can be fibrous and globular. On the basis of nature of molecules, proteins can be acidic and basic. On the basis of their functions, proteins can be structural, contractile, enzymatic, protective, storage, and hormonal. Proteins can serve many functions, like keratin present in hair, nails, etc., become the structural component of our body. You know, when carbohydrates and fats are in short supply in our body, proteins provide energy. Immunoglobulins protect our body from infectious agents. Proteins like actin and myosin present in muscles play a great role in movements of our body. Proteins like hemoglobin, myoglobin, and beta globulin help in transport of materials within our body. Thrombin and fibrinogen work as blood coagulants. Monilin, a protein by nature but much sweeter than sucrose, is used by diabetic patients for their source of sugar. Now I will check how attentive you were in the class. All of you should try and answer the questions before I give you answer. The first question is, what are proteins? The answer is, proteins are polymers of amino acids linked by peptide bonds. Next question is, which scientist derived the term protein from proteose? The answer is, Berzelius and Mulder derived the term protein from proteose word in 1838. The next question is, name the organizational or structural levels of proteins. And the answer is, there are 4 organizational or structural levels of proteins. These are primary structure, secondary structure, tertiary structure, and quaternary structure. The next question is, name the scientist who pioneered the sequence of proteins. The answer is, the scientist who first pioneered the sequence of proteins was Frederick Sanger. The next question is, who found the secondary level of organization of proteins by X-ray diffraction studies? And the answer is Pauling and Corey. My next question is, name the protein in which alpha helical secondary structure is found. Answer, keratin protein, which makes hair, fur, wool, claws, horns, hairs, feathers, etc. The next question is, name 3 types of bonds which stabilize tertiary structure of proteins. And the answer is, these bonds are ionic bond, hydrogen bond, and disulfide bond. The next question is, what is the nature of protein molecules? The answer is, protein molecules are amphoteric in nature. The next question is, what are conjugated proteins? Answer, those proteins which yield alpha amino acid and a non-protein part called prosthetic group on hydrolysis are called conjugated proteins. The next question is, what are globular proteins? And the answer is, globular proteins are proteins in which polypeptide chains coil around themselves to form a spherical structure. My next question is, name one basic protein. The answer is, histone protein associated with DNA falls in the category of basic protein. I am confident that you have learnt enough about structure of proteins, types and functions and enjoyed the lesson as much as I did. Looking forward for the next class. Thank you for your attention and see you next time.